It's my honor now to introduce the second plenary speaker of this year's Max Cohen Summer School, which is Joshua Coon. Professor Joshua Coon is the inaugural holder of the Thomas and Margaret Pyle Chair at the University of Wisconsin-Madison, and there he's a professor of chemistry and biochemical chemistry. He started his academic career in the Central Michigan University, where he obtained his bachelor's, and then moved to the University of Florida to get his PhD. And then later he spent 3 years at the University of Virginia as a postdoc in Donald Hunt's lab, where he developed with John Zucker the electron transfer dissociation fragmentation technique. And then in 2005, he joined the University of Wisconsin-Madison, where he became an associate professor in 2010 and a full professor in 2012. His research focuses on developing new mass spectrometry techniques to study proteins, obviously, and to apply those techniques to advanced biomedical research. To achieve this, he focuses on instrumentation development, data analysis software design, fundamental ion chemical studies, and biological applications of the technology. So a lot of different things. Professor Coon has been awarded many honors and distinctions, and among them, for example, the Wolfram's Faculty Fellowship of the University of Madison in 2014, the Beeman Medal of the American Society of Mass Spectrometry in 2012, the Arthur F. Findays Award for achievements by the young and analytical scientists given by the American Chemical Society in 2011, the Philip R. Certain Dean's Distinguished Faculty Award in 2010, and finally the Kellett Mid-Career Award last year in 2020. So I'm deeply honored to allow you to share your screen now, and I'm looking forward to hearing and seeing your talk.
Thank you for the kind introduction. I thought today I would tell you a little bit about some new things we have for advancing various omics, proteomics and lipidomics in particular. To start, I would just sort of like to called your attention this sort of pathway diagram I'm sure many of you are very familiar with. And while my lab and many others have focused on proteins and proteomics over the last 20 years, you know, I think it's increasingly appreciated that lipids and metabolites are also pretty important. And if you can measure all of these molecules, you can more easily understand the system that you're working at. So today I'll try to show you a couple examples where we apply these multi-omic strategies to learn some new fundamental biochemistry as well. We primarily use hybrid orbit traps. This is an example of one right here. So most of the experiments I'm going to show you today come off of a system like this, where we have a quadrupole mass filter, or we have orbit trap here. And down here we have linear ion traps. And so we find this particular instrument to be very useful for doing peptides, lipids, and small molecules. Because it's a summer school, I thought I would just take the first few minutes to give you a little bit of a background on proteomics, in particular peptide ion dissociation, because that's, I think, very important to understanding some of the things I'm going to show you, but it's just useful to know if it hasn't been covered yet. But briefly, as I'm sure everyone's aware, so far, day 2 of the school, for proteomics, we're going to use enzymes to cut the proteins into smaller pieces. And then we rely on separations to sort out that very complex mixture of peptides. And we ionize, typically using electrospray. And we measure mass. And a mass measurement alone is typically not enough to get sequence, so we need to make it fall apart. That process of making that thing fall apart and measuring the mass of the pieces is called Tandem MS, and you're all learning how to computationally analyze those things, which is fantastic. And you can do quantification based on these signals as well, and then you can do some sort of analytics to see what you found. Now, let's focus a bit on those peptides. And if we think about how we typically do a proteomics experiment, we would use an enzyme like trypsin, which would cleave at lysines and arginines. And so imagine this particular protein sequence, you would end up with peptides that are in various lengths. So you'd have some pieces that are large and some pieces that are small and some that are in the middle. And the typical ideal peptide for our mass spectrometry setup is between probably 7 or 8 and 20 residues long. Things that get much bigger or shorter than that are hard to do. So in any given protein, when we do a trypsin digest, we're going to get a subset of peptides that fall within the viewable window, if you will, of the mass spectrometer technique we're using. But that's okay for most applications because we're just going to need a handful of peptides to serve as proxies to tell us about the presence of this whole protein or even its abundance. So I already went over this figure that shows us the instrument we're using, but let's talk a little bit about the process of looking at and sequencing a subset of these tryptic peptides. So now we've got peptides eluting into the mass spectrometer. These are tryptic peptides. And here I've highlighted 4 of them. And let's say these 4 are being selected for MS-MS. Now, if we go over here in our instrument diagram that we just walked through, all of these ions are going through and they're being mass measured in the Orbitrap, and that's generating the spectrum we see right here. We can then use the quadrupole mass filter to isolate one of those, and then that particular ion will get fragmented. In this case, I'm showing by collisional activation down here in the collision cell. And then those product ions can be mass analyzed to generate our tandem mass spectrum. In this case, that mass analysis happens in the ion trap. You could also do it in the Orbitrap. Each has their pros and cons. But the point is we iterate through this cycle of isolating, fragmenting, isolating, fragmenting. Let's talk a little bit about the tandem mass spectrum. So you all are going to learn or are learning about how to use MaxQuant to do this in an automated way. But just to imagine the way that you can even deduce these spectra by hand, and this is how you would do it. So let's imagine I make this particular peptide fall apart. This particular peak right here corresponds to the mass of lysine being at the N-terminus. And then if I measure the mass between the distance between here and here, that's 71, and that corresponds to the mass of alanine. So now we can introduce the next letter in the sequence. And so you can read the sequence forward with the red ones, right? And you can read the sequence in reverse with the blue ones that come from the C-terminus. These are C and Z ions. You get the same result if you have B and Y ions. We'll talk about that in a moment. All right, so there are many ways to dissociate a peptide in the gas phase. Collisions are by far the most popular, so you collide that peptide ion population with gas atoms or molecules. I'll have a diagram of that in a moment. We can use reactions with electrons. That would be electron capture or transfer dissociation. And we can use excitation with photons, like UV or IR photodissociation. Okay. I always like to put the shark on there. That's just fun. Yeah. Okay. So back to the diagram here. We're going to do this in iteration. Sorry, that one's out of order. Let's go now focus on the collision activated dissociation, which is what most of you will be using. So just wanted to give you a sort of high level cartoon diagram of what's happening. So when you select a precursor ion to fragment, we can imagine that ion being accelerated into this collision cell, which is filled with elevated pressure of gas. And an Orbitrap system is probably nitrogen, and other systems it could be argon or something like that. But basically you're going to accelerate these ions into this collision cell where it will have collisions with the gas. And these collisions are energetic enough that you get vibrational excitation of the peptide and you fall apart into fragments. And these fragments can also have enough energy to fall apart too. Okay, so then the other type of technique, and the data that I'm going to present to you here shortly, we're going to draw on both of these techniques, is the ETD technique here. And in this case, we are storing the peptide precursors that are multiply charged with anions that are singly charged, and you get an electron transfer onto the peptide cation, and that causes a backbone cleavage that is not vibrationally motivated, but rather driven by free radical chemistry. So the reason we like these types of methods is that for certain types of peptides, things that have very labile modifications, things that are not tryptic peptides because they're either very large or they don't have basic residues on the end, or even whole proteins, these ETD technique can be very good for. Okay, so now I'm going to tell you enough with the tutorial stuff. Let's go into some examples. So I want to share with you a few projects. The first one is a project to deep sequence the human proteome. And this has been a collaboration with Jürgen and Pavel from Jürgen's group and Alicia from my lab. So basically, as I taught you in the beginning, when we do proteomics, we typically measure the presence or absence of a protein with only a handful of proxy peptides. So if you take a human cell line and you do digestion of that, and you spend 10 to 24 hours analysis on the mass spectrometer, you can identify well over 10,000 proteins. But if you go look at like how much of each protein do we detect, in this heat map, each pixel represents a protein of these 10,000 we detected. The really dark color indicates that we've covered most of the amino acids in that protein, whereas the red indicates we've covered less than 25% of that protein. And you can see the majority of the proteins, we don't detect most of them. And that's fine for quantification, but if you really want to characterize the proteome, you'd like to be able to cover every amino acid of every protein. So one way to address this particular problem is to explore the use of other proteases aside from trypsin to get more coverage. Doesn't mean we want to discard trypsin, but we need to add to it. So here's an example. If we take that protein we started with, here you can see if we digest with trypsin, we get a couple peptides of suitable size. We digest with a different enzyme, say GluC, cleaves at glutamic acid here, you can see that we get different peptides in our size window, and now we can boost our coverage a bit. Imagine adding 3, 4, or 5 other enzymes. We can then boost the coverage by bringing different portions of the protein in and out of our visible window. We can also use different dissociation methods, both collision and electron transfer driven dissociation. So here's an example of a peptide from yeast, which if we digest the yeast proteome with lysand and do HCD activation, we detect all of these peptides shown in blue. So we get about 40% of the protein covered. That's pretty good. Here's a spectrum of one of the peptides. Now, if I add and I do a separate experiment where I do ETD on the peptides, I can get different parts, some overlap, some different. Here's a new piece. And now I've boosted coverage to 63%. If I use both dissociation techniques and about 6 enzymes, I can cover just about every amino acid in that particular protein. So it's very tedious, but if you want to get high coverage, it's a way to do that. So we set out an experiment to see if we could sequence fully every expressed protein. Now, we probably can't get that goal, but we can try our best. To do this, we have grown 6 human cell lines in culture. We harvest proteins from them separately. We digest those proteomes with 6 different enzymes separately. And then we fractionate and do LC-MS with both ETD and HCD for a total of about 2,500 LC-MS experiments. So that was a lot of work. You wouldn't do this every day, but if you want to see what the limits of our technology are, it's very exciting to do that and spend that energy. In total, we provide evidence for almost 18,000 expressed proteins collectively in those 6 cell lines, with a median sequence coverage of close to 80% in human cell lines. So we were very excited about that. This diagram shows us for each cell line how many proteins we detected as a function of which protease we used. And then this is cumulative. And you can see trypsin is red. It always does the best. And that's why we use it as our go-to enzyme. But you can see these other enzymes contribute a pretty good amount of information. And that information is different and in many cases. So in total, we have about 18,000 protein groups representing about 12,000 genes. And about 800 of our proteins have 100% sequence coverage. So now we have a pretty unique proteome data set that we can ask questions, fundamental questions about what's going on in the proteome and what are some basic processes that happen. Can we assess these? This is a diagram, again, to sort of showcase the different coverage we have. And you can see most of the proteins have very high coverage, and then you can see trails off. We don't have very many with low coverage. Just to put this particular data in perspective with other published works, the human draft maps are down here in terms of the blue represents the number of unique peptides. And the study by Nuno's group here was the largest published to date. Ours isn't quite published yet, but hopefully soon. But you can see we have a tremendous number of peptide spectral matches and unique peptides, about 1.1 million here. All right, so now we can find evidence for new things, or that's our hope. And here's just an example of 3 distinct isoforms that, that are all the same except for the C-terminus. And you can see there's different ways to end this protein, and that gives you the rise to 3 isoforms. And you have to detect these peptides on the C-terminus to know which you have. And it turns out we can detect peptides from all 3, so we can confirm that all 3 of these isoforms are actually expressed in these cell lines. Now, this is where Pavel and Jürgen have been really working hard on this data. And here's so we can take that idea of looking at isoforms and say, could we do a global analysis of alternative splicing? So this is a high-level characterization of different types of splicing you could get: alternative acceptor, alternative donor, retained intron, and so on. You probably read about these in your textbooks. But basically, there's different types of splicing that can happen, and nobody's really been able to assess at the protein level how much of these RNAs that get spliced actually make it into functional or viable protein that is expressed in the cell. So here's how this would show up in a sort of a cartoon diagram on a particular protein, amyloid precursor protein. So here's a case where we know from the RNA data that you can have exon skipping. So exon 8 can be skipped. So we know there's transcript evidence for this form where you have exon 8 missing, and then there's transcript evidence where it's not skipped. So if you want to know if which or both of these proteins are present, you need to detect peptides that span this junction between exon 7 and 9, and you would need to detect peptides that span 7 and 8 and 8 and 9, so these peptides here. From our data, we have 3 unique peptides coming from that junction, and several coming from this junction, and 2 coming from this junction. So we can say with confidence that both of these protein splice variants are present in these cell lines at the protein level. Oh, sorry, I went a little ahead of myself. Here's just some tandem mass spectra that showcase a couple of these particular peptides that were detected. So now the question is, to what extent do these splicing events happen on a global scale? So here's some data that Pavel's put together on looking at, as a function of enzyme, how many peptides do we have per enzyme that either tell us about, areas where there are no junctions, that's the green, that's the majority, areas that cover one junction, and areas that cover more than one junction. And there's not too many of those, but there are a significant number here that cover one junction. And you can see, as a function of enzymes, these guys add a lot. And you can see that as a percentage, you get slightly more from these other enzymes, whereas the trypsin's a little bit lower. And the reason for that can be seen in these next 2 plots here. And so this shows us the distribution of peptides that don't cover a junction, but where they are with respect to a junction on the N or the C term in distance of nucleotides. And what we can see here as a function of enzymes, so the enzymes are down here. So you can see here there's this bump at 0 for trypsin and lyse C on both ends of this, and I think I can explain this better on the next slide. So if we go to this slide, what we'll see here is, now this is looking at peptides that span a junction, and what you can see here is that 3 nucleotides in from this junction, you have a spike in lyse N, and on the C side we have at one nucleotide in trypsin. And that's because it's very interesting and neat to see, because it turns out that these junction regions are enriched for nucleotides that code for lysine and arginine. And this has been noted in a recent publication that if you want to look at alternative splicing on a large scale, it's limited by trypsin cleavage specificity. And the upshot of that work is that the nucleotides approaching these junctions are enriched for three-letter codes that code for either lysine or arginine, which is where trypsin cleaves. And so you have a high probability of having one of those residues there, so that you don't actually get a peptide that spans that junction. So that's why we see these spikes for lyse N on one side and trypsin on the other. And the other enzymes don't show that so much. So the point is, by having many different enzymes, we have a better chance, we can expand our chances of hitting these junctions. So then I will share with you sort of the take-home message, which is now we have a global map of to what extent does splicing happen at the protein level in these different categories. So here we have what you'd predict from the genome annotation. Here's what you see at the transcript level, and here's what you see at the protein level for skipped exons. Alternative, these are different types, and you can see most of them go pretty close to the transcript level at the protein, except for retained introns, which we don't really find evidence of. And that had been noted one other time in the literature, so that is in agreement with what we see. So we're still mining this and we're still putting the story together, but it's really exciting to be able to see globally what's going on at the protein level in a new way. And that's really been driven by changing up the methodology and leveraging different technologies to access these peptides that are hard to get to. All right, so I want to tell you a brief now technology story and regarding TMT quantitation, which I'm sure you've already heard a bit about in yesterday's plenary lecture, and how we can do a little better job with TMT. So this work isn't published yet either, so you're getting a sneak peek here, but in Professor Gigi's lecture, he probably talked about this method called Multinotch MS3 for TMT labeling. So the idea is that when you TMT label a peptide, you have these reporter tags on the fragments, and if you want to get the best signal possible, you isolate these Y ions, and then you break them up, and you release the reporter ion from them, and that gives you the best quantitative signal that you can get for these reporters. But the price of that is that you reduce signal, because you're throwing away most of these things and you're only picking a handful of them. So your signal noise on these reporter ions can be low. Okay, so we've taken a look at that, and it turns out that's the case. So here's what happens if you take a B ion, shown here the B9, and we fragment the B9 with HCD, collisional energy. The best we can do at converting that signal into the reporter ion, which we need for quantitation, is about 22% conversion efficiency. And you can see down here, if I look at as a function of collision energy, right, we can only set one collision energy, so we hope to pick the best one. If you pick 65 volts, you get the 22%. But if you pick off of that, you're going to get even lower conversion efficiency. Now, if you go over here and you look at the B5 ion, it's a higher M over Z, it's got less charge. You can actually get 43% of the TMT release. So you can get an efficiency of 43%, but you need a voltage of 100 volts to do that. And if you were to run it at 50, like the other one wanted, you'd have a much lower efficiency. Now, so all of these fragment ions, they have different optimal energies for converting the fragment into the reporter ion, which you want for quantitation. So we had this idea that maybe, so this is kind of a little bit exaggerated. It don't really have fire coming out of our mass spectrometer, but in HCD, we are making the thing fall apart by collisions in this region. We have a different way to make peptides fall apart, and that's using photons. I introduced this earlier. We can just shoot a laser beam. I should have put the shark here. We can just shoot a laser beam down the axis of our device, and we can basically think of sort of melting these things. We can slowly cook them with this IR energy and pop off the TMT tag. And we can do them all at the same time that way. And so now the concept is, if we take this multi-notch idea, we fragment this, we get this spectrum, we pick these fragments for then isolation and converting into TMT. If we use our IRMPD process, you can see as I turn up the power, they all get better. And then when I hit max power here, they're all just like, that's the best, highest efficiency I can get at converting these into reporter. If I did the same experiment with HCD, as I showed you earlier, I get a wide distribution of optimal energies, but I can only pick one. So basically, if we do this experiment on a large scale, this is comparing the ratio of IRMPD to HCD produced reporter ions if we isolate 10 fragments in this method, Multinotch method. So you can see I can boost my signal to noise by 1.6%. So a pretty good bump in the signal. And as I choose more precursors to dissociate, I can slightly improve that up to 1.7, down from 1.5 if I only choose 2. That's because as I add more, I get more and more range of optimal HCD energy, so you get more and more of a bump. So the upshot of that is here, that if I do a sample where I'm limited, I don't have a lot of material, like single cell you're going to hear about later this week, you don't have a lot of signal to work with, so you're always fighting low signal noise. So here's a mixed TMT ratio, and we're just asking, of the things I make fall apart, how many of them give me TMT ratios above some defined threshold, signal to noise? It's only about half if I use HCD for this approach. But if I use the IRMPD approach, I can boost signal, and I can get 80 to 90% of these things up above that threshold. So I'm boosting signal. So it's a neat trick. It's not going to change the world, but it's an interesting trick that can boost signal to noise, particularly for low-level applications. All right, so that's some technology. Let's talk about some biology and how we can apply these techniques to learn something new functionally about how proteins work. So this is a project led in my lab by Evgenia Shishkova and Katie Overmeyer, and in collaboration with Dave Pagliarini's group, who just recently moved to Washington University. All right, so the idea here is that many mitochondrial proteins are poorly characterized. This little chart shows us that, like 50% of mitochondrial proteins, have only 6% of the publication. So like, there's some superstar mitochondrial proteins that everybody studies, and most of them, nobody knows what they do, or many of them. There are many mitochondrial pathways that have very limited annotation, so we don't necessarily even know who the players are. And there are many mitochondrial diseases that we don't even know why they're happening, and there are no proven effective therapeutics for mitochondrial diseases. So the concept of this particular project is that if we have a collection of human cell lines that are each missing a different gene, we could then learn something about them. So basically what we do is we have all these different mitochondrial pathways shown by these cartoons up here, and we select genes from each of these pathways to knock out. They're called our sentinel targets. So we basically have 113 cell lines where we knock out a sentinel gene, which we know which pathway it's in and we know its function. Then we have 90 cell lines where we knock out a gene of unknown function, but we know it's mitochondrial. We do replicates of each, 3 to 4 replicates. We have about 800 different cell lines to process, but we're going to do the proteome, the lipidome, and the metabolome on each of those. So 2,400 seems to be the magic number for large-scale studies in my lab, I guess. So anyhow, now we have, we've analyzed these 800 cell cultures. We have about 8 million biomolecule measurements. We cover about 8,400 proteins, 3,500 lipids, and 160 metabolites, and we can, put this out. We can make this giant heat map you're seeing over here. About 12,000 molecules quantified per sample. But let's talk about what you can do with it. So here's an interesting example of this pathway. So here's this pathway from one of these pathway maps, and what we can see here is that this particular gene, ALDH18A, which is this guy, catalyzes the production of proline. And now this plot shows us the abundance of metabolites if I knock out that particular protein in comparison to wild type. And what you can see here is that proline is down, is down-regulated here. And that's because it's in this particular pathway. If we take this away, we don't make proline as effectively as we did. So we see a big reduction in proline. So that's pretty cool. But now what's very interesting is that if we now ask, okay, let's just look at the abundance of proline. So now every dot is showing us the abundance of proline in a different knockout cell line. And we can see these ALDH, 2 different knockouts are both downregulating proline, which we would expect, but we also see this gene NADK2, knockout 1 and knockout 2, both also showing us a downregulation in proline. And so now we can assume that NADK2 might have something to do with this particular pathway. And I don't think I show it here, but there was a paper that came out earlier this year that was like a whole paper, and it just said that. And it was just in our data for a couple of years, and we didn't notice it until that paper came out. So that's the importance of looking at data. But it's right there to find. So here's another example. This particular gene, CPT2, catalyzes acylcarnitine conversion to carnitine. And if we take it away, compared to wild type, every dot now is a different lipid. We can see an accumulation of acylcarnitines because they're not being processed with this particular enzyme. All right, let's learn something new. So here's a plot. We can just look at our data in all these different ways. We can say, show me the abundance of a molecule. In this case, we're going to be interested in CoQ10. CoQ10 is an important cofactor. And all of these genes, if we take them away, and as their name implies, they are involved in CoQ synthesis, all except for this one, we see downregulation in CoQ10 if we take those genes away. Now we can just ask, are there any other genes that also cause a downregulation in CoQ abundance? So these are all the different gene knockouts. And this one here, PIRF, actually does that. So now we just look at CoQ abundance, and these are all different knockout lines. We can see that PIRF, along with the CoQs, if we take it away, causes a downregulation in that particular molecule. And so now we can go to the proteomic data. We just sort of flip, and we can say, okay, what are the proteins doing when we take away PIRF versus wild type? And we can see that PIRF goes down as we'd expect because we knocked it out. But these NDUFs, these CoQs, and these other NDUFs, which are all, it turns out, if you pull down PIRF, you pull down these 2 guys here. And if you pull down this guy, these are interacting over here. So they're all previously known protein-protein interactors. And now we have evidence that are involved in CoQ synthesis. And I won't go into the motifs here of binding because I'm running short on time and I wanted to tell you more. But the point is we can now connect PIRF to CoQ synthesis and its involvement in this complex and stabilizing it. And now we ask, can we find a human disease where this particular gene is involved? And so it turns out that we've identified a patient that has a homozygous mutation in PIRF, and they have all of these issues. Unfortunately, the patient passed away before we were able to make this connection, but presumably a CoQ deficiency was underlying all of this because of a mutation in PIRF. If we knock out PIRF, we can cause loss of function. We can cause gain of function by knocking in the patient mutation. We've done all sorts of things. I won't walk you through all that. But the point is, we've now connected this particular gene to a function, and we've got about 6 or 7 others in the study that we're finishing up right now. So that's a way you can leverage these multi-omic studies to learn some new biochemistry. All right, I got about 10 minutes to share with you sort of the last multi-omic part of this technology that I'd like to get into. So let's talk a little bit about lipids. So lipids are kind of like peptide sequencing, just a little bit different. You don't have to digest them, obviously. But basically, we do C18 separation of lipids. We measure their mass. We do MS2 to get their identity. So it's very similar. I'll talk a little bit about, if you're not familiar with lipids, they have conserved head groups. So you can have head groups. You can have this connector here. And then you have varying length chains that go on them. And those chains can be saturated or unsaturated, and they have these double bonds. And so localizing the double bonds. So you want to know the chain length, you want to know where the double bonds are, and you want to know how it's connected and what kind of lipid. That defines the lipid class. So basically, name of the game here is to know the type of lipid that's defined by the head group, to know the chain lengths and to know the double bond number and to know where the double bonds are. That's what you'd like to know. So we'll skip that in the interest of time, and we'll skip the extraction other than to say, we do separations on them, and we do high resolution MS1 that can help us learn what class that they're in. And then we do tandem MS to get the chain length and hopefully the number where the double bonds are. All right, so it's very similar to peptide mass spec in that way. So this fellow in my group, Paul Hutchins, wrote some tools to look at lipid data we call Lipid X. I'm not going to go into the details of that, but basically it helps us determine which lipids are present and all of this information I just told you about by sort of disentangling some of these and interpreting tandem mass spectra. Okay, we're going to skip this project in the interest of time here and go straight to this one. Okay, so Thermo has introduced a new mass spectrometer called the IQX that we've been helping develop. And in this case, so most of our lipidomics work had been done on a Q-Exactive platform. So this platform has a quadrupole mass filter, a collision cell, and an Orbitrap. So for all of the analysis, we're doing high resolution, so MS1 and MS2. The Tribed platform shown over here has the quadrupole mass filter, the collision cell, and the Orbitrap, but it also has these ion traps down here. And what we wanted to know is, can we use these ion traps to really accelerate how we're doing lipid ID and analysis? The ion traps are pretty cool because they allow really fast scanning and they allow MS to the N, which means we can sort of really take apart a lipid in a systematic way very quickly by using multiple rounds of tandem mass spectrometry. The system's also capable of real-time decision-making, so we can actually look at a spectrum, decide what to do based on the results of that spectrum, and change our next decision. So this device has what's called the real-time library search for intelligent mass spec of small molecules. So basically what you can do is, as I mentioned, you can take an MS2, you can search it on board system, you can get a result, and based on that result, you can dictate how you want to run the mass spectrometer next. And I'll show you some examples of why we think this is going to be pretty useful. So Dane and Yu-Chen in my lab have been working out this, and this has been a collaboration with the folks at Thermo Fisher. So it's been a big team effort. All right, so here's one particular problem that I didn't yet mention with lipidomics, and that is that unlike peptide mass spectra, you get what we call degenerate MS1, and that can limit sampling depth. So let me explain. When you ionize by electrospray lipids, you tend to get adducts. So you get lipids that have ammonia, sodium, potassium on there, and you also get dimers. So you get two lipids together, and then those dimers will have these adducts. So then you'll have the peak for the ammonia, the peak for the sodium, the peak for the potassium. You also tend to get in-source fragments. So sometimes the head group might fall off or something might fall off. So it turns out in this particular case, we have about 10 unique mass to charge peaks in the MS1, but they're all coming from one lipid species. So that means if I'm saying, sample the top 10 most abundant things, I might only pick the same thing 10 times. Like, that's really not helpful in getting good depth. So ideally, we would pick one of these peaks, we not pick the rest of them. Well, I suppose super ideally, we'd only make one peak, but I don't know how to do that. So we're just going to try to avoid the ones that we don't want. So here's an example of this cholesterol ester. It has multiple peaks here, as I can show you, just like the other one. And what we can do is we look at all these different classes of lipids, and we can say, it wasn't just that one, but you get dimers and adducts happening. These are all these different lines. So for PE or triacylglycerides, we see this. We see this for pretty much all of these lipids. We see multiple different adducts and dimers. That's a problem. All right. So now we've set up the system so that it can do a real-time search, and it can ask the question, once it identifies something, it calculates all of the sodium and the dimer peaks. And if we turn that on, we can now increase the number of lipids we identify because we're avoiding sampling sodiums and dimers. Now, if we tell it don't sample sodiumatics or dimers or in-source fragments, we can see another boost. So we're still figuring out the best way to run this, but already these are very early data. We're showing that if we can avoid selecting these degenerate features, we can sample deeper into the sample set. So that's been pretty exciting. So here's there's some published results of how real-time, that if you had the ability to make real-time decisions, you could do good things. So here's a case that where this particular thing, when you do MS2, you get a mixture of neutral losses. And if you can trigger an MS3 event, you can actually figure out exactly chain lengths and which isomers are present. But you have to be able to do MS to the 3 in this case. This would require the ion trap. That was done manually, by the way. Here's a case where you have ion trap collisional activation producing a much better spectrum than an HCD spectrum. I didn't go into ion trap CAD, but it's a bit different than beam type HCD. So it produces a different spectrum. So here you can see it's beneficial to use a different type of collisional activation. And then I'm going to skip this one in the interest of time so we can wrap up here and just say, here we have human plasma. This is data we generated. We have an MS1 where we pick this lipid, we fragment the lipid, and we get an Orbitrap MS2. And the real-time search tells us that we have a class identification. And if we triggered an MS to the N, we would be able to enhance that and learn much more. And so we do, and we get chain length now. We learn it's a 16.0 and an 18.2 based on that triggered ion trap MS to the 3. Here's another one where the ion trap MS2 scan was mapped, and we triggered an autonomous NS to the end that gives us degree of the length of the chain and tells us this has one double bond on it. I'm going to skip this because it's very much the same. And so lastly, I would just say in the few minutes we have left that we're now also, most lipids, I failed to mention in the beginning, are separated at high flows using more traditional, microliter flow rates, not capillary flow rates like you do with proteomics. And we want to change that. We want to bring everything down to a capillary LC column. So we've been working on doing lipidomics with nano LC, and I'll show you why we want to do that in a moment. And I'm just basically hitting the highlights to say that we've been very impressed with capillary LC. This is a separation we've done with capillary LC on lipids, where we get over 1,000 identifications from plasma on an Orbitrap Eclipse platform. We can do ion trap MS2s, and we get very comparable, the spectra are the same as you'd expect, just lower resolution. But when you look at cumulative IDs across a gradient, you can see the ion trap, because of its scan speed, outperforms the FTMS, which is cool. So now I will explain why we think we want to go to a unified platform. So the idea here is that for our roadmap for how we would develop multi-omics is that right now for that, that mitochondrial project I showed you, we had 3 different mass spectrometers. We had 1 sample coming in, we split it 3 ways. We had 3 analysts, 3 mass spectrometers generating 3 types of data. Seems kind of wasteful. The same mass spectrometer can measure proteins and lipids and metabolites, but we have it all split up. So ultimately we want to be over here. We want one sample prep method that can give us metabolites, lipids, and peptides, put it on 1 mass spectrometer, and out comes all of these ohms, 1 analysis. And MaxQuant will by then be able to analyze all those in 1 shot. And that will be really useful for being able to go from looking at tissues and samples where you have tons of cells to looking at single cells. Imagine you put the single cell at the head of the column, you crack it open, all of the contents go on, and we have 1 method and we see everything. You can't look at the proteins on a separate instrument if you've got the single cell, right? It's all going to have to be done together. So we've been developing this method we call Multi-Omic SingleShot Technology. led by Yu Chen and Katie in my lab. And so here's the cover here. So we've got these lipids that look like teeth to me, and peptides all in the same column, and they just come out separately and they go into the Orbitrap. And I've just promised this is the second to last slide, or maybe the last slide. We have the gradient set up so they're both lipids and proteins on the same column. The peptides come out, gradient kicks up a notch to stronger mobile phase strength, and then the lipids come out. This is on a capillary LC column. It's hard to tell from here, but if we have the lipids on the column, we get 5,000 proteins detected. If we do the proteins separate without the lipids, we get the same thing. The lipids do not seem to be affected by the presence of the proteins either. So in the end, we can have them both there. And just by extending that gradient, we can get both proteins and lipids from the same sample. Now we're working on getting metabolites in, which we'll probably allude up here, but we're working on that. So that's where we want to go. This is my group. I pointed out most of the people that did this work as we went along, so I won't do it again here, but I would just like to thank our funding agencies. Most of our funding for these technologies comes from the National Institutes of Health. We also have a partnership with Thermo Fisher, which we're very grateful for. And I want to thank all of you for tuning in today. It's about 1.1 thousand folks, so that's one of the bigger audiences I've ever spoken to. Although I can't see you, it's been a pleasure to be here. Thank you.
Thanks, George, for the great talk. It was really nice. One quick question. So you showed that you are developing this peak picking avoidance for the redundant lipid species there. So are you doing, or is somebody developing something similar for metabolomics? I guess the same problem there. And,
yeah. Pardon me. Yes, the real-time search that the Thermo team has been developing is also been developed for the metabolites as well, but our team was tasked to help them on the lipid side, but I know that they're working on metabolites too. So many of the same concepts are being applied there. I just don't have those examples because we focused on lipids.
So thank you very much. This was really great. Firework of information for me personally. So I would now like to give some of the attendees the option to ask questions.
In the beginning, you showed that HTD is like the energy for good fragmentation is very different for the different TMTs. Can you explain how this, where this is coming from?
Sure. So basically, if you were to, so imagine when we're doing HCD, you're taking a population of ions and you're accelerating them into a chamber that is slightly pressurized relative to where they were coming from with gas. And so they're going to be having collisions with that gas. So, and the voltage that you apply is basically a proxy for how much they're being accelerated, right? So the bigger the number, the faster they're going in there and the harder they're hitting. So each peptide will have an optimal collision energy. That is defined by, I would say the optimum is when you get the best signal and diversity of fragments, right? And that is going to be a function of the peptide's mass to charge, the peptide's charge, and to some degree, the peptide's amino acid sequence and where the charges are on the particular peptide. So you will see a range of optimal sizes. Now, the devices are, so when you're doing an MS2 of a selected precursor, that can all be calibrated in. So you know the charge, you know the mass to charge, you don't know the sequence, but you know those two things, which are really high drivers. And you can get your, the instrument will automatically, try to help you get the right HCD energy and maybe use multiple energies. But if you have a wide diversity of fragments, like in the multi-notch experiment that you're making fall apart, you've got to do them all at the same time, but they're very disparate in their mass to charge and potentially their charge. So that's where it becomes a compromise and particularly problematic. So it's not the typical experiment, but that experiment in itself is sort of really the worst case scenario where you have to deal with that. I suppose DIA is somewhere in between because you are selecting a window which is comparable in mass to charge, but you may have lots of different charge state things in there. So that's somewhere in between.
Thanks for the insightful talk. I was just wondering, you were talking about combining the proteomics and metabolomics and maybe the lipidomics together in the single cell. But I just wondering, like proteomics, have the research combining with the TMT tags, and how would you comment on just gather enough signal from the metabolomics or the lipidomics?
Oh, sure. Well, I would just clarify, I'm not working on single cell. It's an aspirational example, I would say. But I agree, many of the folks doing single cell are using TMT, and in the scenario I just described, that's really not compatible because I don't really know how you would also label the metabolites and the lipids. So I suppose the way I have sort of visualized it sort of excludes that approach. But in my mind, this is sort of like, if one could make this work well, that would be an exciting application of its technology, although I don't know how it's exactly compatible with any tagging at this moment. So yeah, thanks for the clarification.
Cool. So for the sake of time, we'll take one more question, which will come from Olga.
Hello. First of all, thank you for very impressive talk. I have a question to the second part of your talk, where you're able to resolve proteins and lipids. So first question is regarding sample preparation, how you achieve this. And second question is, so how do you plan to integrate metabolite in the game? So if I understand you correctly, you resolve lipids and proteins in one gradient, right? That's correct. Yes. So right now you're absolutely correct. So it's been rather straightforward to distinguish to separate the peptides from the lipids based on just their behavior. So it turns out just by luck that the peptides, when they stop eluting, that's when the lipids start, and so it's really very convenient. And then I think you asked me a question about how we did sample prep. So right now, for the data I've shown you and the data we have published, those are prepped in the traditional way and then loaded on one after the other. I have a group of people working on sample prep and extraction and loading where we can do that together. It's not quite done yet, but it's promising, and we think there are ways to do it. Other people have thought about this before. There is some literature precedence. So it seems quite possible. Now, to your last question about metabolites, that is the next sort of step in this project, now that we've moved to capillaries and we've got that working for lipids and peptides. So what we're looking at for metabolites is that, things like we might have to look at having different stationary phases, at least in part on the front of the column. We may have to look at cooling down the column a bit to retain those early and shelling out that gradient. So we've recruited a friend of mine, Bob Kennedy, who's a great chromatographer, and it's going to be mostly his problem to figure that out. But we think it's possible, but it's more challenging. Now, if the metabolites and the peptides are co-eluting, I don't see that as a big problem, because I think we can train the mass spectrometer to know how to behave and to deal with that. So the co-elution, I don't think is a big deal. I think we can totally work around it, but getting them retained and separated is probably the bigger challenge, which we are about to get started on. So I hope we're successful. We'll see. Thank you for that question.